The concept of antibiosis and selective toxicity are fundamental principles in antimicrobial therapy. Antibiosis refers to the antagonism between organisms, where one inhibits or destroys another. This phenomenon was observed even before Fleming's discovery. When a mold or fungus produces compounds that inhibit bacteria nearby, we can observe a zone of inhibition where bacteria cannot grow. This natural antagonism between microbes was known as antibiosis, a term describing how certain organisms can inhibit the growth or survival of others. Fleming's key contribution was demonstrating selective toxicity. Penicillin could inhibit certain bacteria while leaving others completely unharmed. This selective action happens because penicillin targets specific structures in the cell walls of some bacteria, but not others. The bacteria that are affected are those sensitive to the antibiotic. This principle of targeting specific pathogens without harming the host became a cornerstone of antimicrobial therapy. It allows us to treat infections while minimizing damage to human cells. Selective toxicity is what distinguishes effective antimicrobials from general toxins. Unlike chemicals that destroy all cells indiscriminately, antibiotics like penicillin target specific bacterial structures. Fleming's demonstration of selective toxicity revolutionized the approach to fighting infections and laid the groundwork for modern antimicrobial therapy. Penicillin revolutionized antimicrobial therapy, transforming the treatment of bacterial infections that were previously deadly and often fatal. Before antibiotics, even minor infections could lead to death. Penicillin saved millions of lives worldwide and changed the course of modern medicine. Fleming's work marked a critical shift from broad-spectrum antiseptics to targeted antimicrobial agents. Antiseptics were chemicals that killed both harmful bacteria and human cells, limiting their effectiveness and causing damage to the patient. In contrast, antibiotics like penicillin demonstrate selective toxicity, targeting bacterial cells while leaving human cells unharmed, making them much more effective treatments. Fleming's work also led to the development of differential culture media, a critical advancement that allowed scientists to isolate and study specific microbes. By incorporating penicillin into culture media, scientists could selectively inhibit the growth of certain bacteria while allowing others to thrive, enabling more precise microbial identification and research. Fleming's antimicrobial assay methods revolutionized laboratory science. In its simplest form, Fleming's assay involved growing bacteria on a plate and observing areas where their growth was inhibited by antibiotics. These methods evolved significantly over time, becoming more standardized and sophisticated. Modern assays use microtiter plates that allow for testing multiple antibiotics at different concentrations simultaneously. Key advancements in antimicrobial assays transformed how we evaluate antibiotics. These laboratory methods had profound impacts on antibiotic development and our understanding of bacterial behavior. These foundational assay methods would soon evolve into more specialized techniques like the disk diffusion method. The disk diffusion method became the standard for antimicrobial susceptibility testing in clinical laboratories during the 1950s. In this method, filter paper discs impregnated with antibiotics are placed on an agar plate with a bacterial lawn. As the antibiotics diffuse into the agar, they inhibit bacterial growth, creating clear zones around the discs. The Kirby-Bauer assay standardized this test with specific parameters to ensure consistent and reliable results. Results are interpreted by measuring the diameter of the inhibition zones and comparing to standardized breakpoints. Over time, the disk diffusion method has evolved from manual testing to increasingly automated systems. Modern automated systems integrate computers for online data analysis, improving efficiency and accuracy. These systems offer numerous benefits, including increased throughput, reduced human error, and network integration for data sharing across healthcare facilities. Rapid molecular methods have revolutionized microbiology by dramatically reducing detection time. These techniques, like PCR amplification, can detect organisms and antimicrobial resistance genes within just 1 to 4 hours. This represents a significant improvement over traditional culture methods that typically require 24 to 48 hours. Another important innovation is chromogenic culture media, which facilitates direct detection of multi-drug resistant organisms. These special media contain chromogenic substrates that produce visible color changes when specific enzymes from target bacteria are present. Results from chromogenic media are typically available within 18 to 24 hours, still faster than traditional methods. The key advantages of these rapid methods include faster diagnosis, earlier appropriate treatment, reduced inappropriate antibiotic use, and improved patient outcomes. Together, these advanced laboratory methods are powerful tools in the fight against antimicrobial resistance. Current research focuses on developing rapid diagnostic methods and genotypic resistance testing to combat antimicrobial resistance. Modern diagnostic methods allow for faster identification of pathogens and detection of resistance genes, often providing results in hours rather than days. DNA-based assays use techniques like PCR amplification and microarray technology to detect antibiotic resistance at the genetic level. These assays can identify specific resistance genes, such as those for beta-lactamases, carbapenemases, and methicillin resistance. Whole genome sequencing has become a readily available tool for precisely detecting antimicrobial resistance. WGS provides complete genomic information, allowing researchers to track the evolution of resistance and identify novel resistance mechanisms. Compared to traditional methods, these molecular approaches offer higher sensitivity and specificity and can predict phenotypic resistance before it manifests clinically. These technologies have critical applications in hospital outbreak investigations, surveillance of emerging resistance, and guiding targeted antimicrobial therapy. As these technologies continue to advance, they're becoming faster, more accessible, and increasingly integrated into clinical practice to combat antimicrobial resistance. Antimicrobial stewardship programs, or ASPs, are coordinated interventions designed to promote appropriate antimicrobial use in healthcare settings. These programs include several key components that work together to optimize antimicrobial prescribing. When properly implemented, ASPs significantly reduce inappropriate antibiotic prescribing, decrease antimicrobial resistance, and improve patient outcomes. Complementing these programs are combination therapies, which represent a strategic approach to antimicrobial treatment. Combination therapies involve pairing two or more antimicrobial agents that work through different mechanisms to target bacteria more effectively. These combinations offer several advantages over monotherapy, including enhanced efficacy and significantly reduced risk of resistance development. Several combinations have proven highly effective in clinical practice. For example, combining a beta-lactam antibiotic with a beta-lactamase inhibitor restores efficacy against resistant bacteria. Together, antimicrobial stewardship programs and combination therapies represent crucial strategies in our ongoing battle against antimicrobial resistance, helping to preserve the effectiveness of our antibiotic arsenal. The future of antimicrobial therapy involves innovative approaches to combat resistant pathogens. Personalized medicine is emerging as a promising approach, tailoring treatments based on individual patient genomics and specific pathogen characteristics. Several alternative therapeutic approaches are being developed to address antimicrobial resistance. These include bacteriophage therapy, using viruses that target specific bacteria, antimicrobial peptides that mimic natural defense mechanisms, nanotechnology for targeted drug delivery, and CRISPR-based antimicrobials for precise genetic targeting. Addressing antimicrobial resistance requires a comprehensive global approach with coordinated efforts. This includes international research collaboration, strengthened infection prevention measures, responsible antibiotic use, and integrated surveillance systems to monitor resistance patterns. Combating antimicrobial resistance requires a multifaceted approach, combining innovative therapies with responsible global practices.